Okay, so this video is on AP Bio topic 4.5, feedback, and I'll be focusing on negative feedback loops in this video, and then my next video will focus on positive feedback loops. So when we look at feedback loops and what their purpose is, it really comes down to how our body is going to maintain homeostasis. So homeostasis is how our body maintains an internal stable environment. And so things that could possibly change in humans could be their water level, depending on how much they're sweating or drinking, etc. Or it could be the blood sugar levels, depending on when they've last eaten. And how much carbohydrates they ate or how much activity they're doing. We also have our blood pH to maintain at close to 7.2 pH, certain ion concentrations like calcium levels within our blood, as well as our body temperature, maintaining it at 37 degrees Celsius. So when we look at the human body and we look at all of the struggles that we face on a daily basis, we have both external as well as internal changing environments that we're constantly working towards maintaining at a constant state or as near a constant state as possible. So sometimes our body temperature might go up, but then we're going to work through homeostatic mechanisms to bring it back down. Or if our blood sugar levels rise, we have a way to bring it back down, or if they drop, we can bring them back up. And so that's what we're going to talk about. So how do we maintain homeostasis though? So we rely a lot on the endocrine system to help maintain our internal conditions. Now, we will also use the nervous system as well as the excretory system and a lot of different body systems all working together. But in this video, we're going to focus on our hormones and the endocrine system. So the endocrine system is composed of organs that are called glands. And glands are organs that release hormones into the bloodstream. So you may hear two different terms. You have endocrine glands as well as exocrine glands. So endocrine are ductless, and then exocrine would be like a sweat gland where like there's like an actual tube to deliver the sweat to the surface. So a sweat gland is not the same as an endocrine gland. Okay, so some major glands within our endocrine system are the anterior and posterior pituitary. Arguably, the anterior pituitary is one of the most important parts of the endocrine system because it connects the brain, the nervous system, with the endocrine system. The brain hypothalamus will actually receive input about conditions, both internal and external, and then communicate with the anterior pituitary to then send messages to the rest of the endocrine system. Endocrine system to help us maintain those constant conditions. Now, this is also tied with the circulatory system because these hormones are going to travel throughout our bloodstream. So anyway, we have the anterior and posterior pituitary glands. We have the thyroid gland in charge of metabolism. We have our adrenal glands on top of our kidneys, our pancreas, and our ovaries, and our testicles. And so glands throughout our endocrine system release hormones, and hormones will travel through the bloodstream to reach their target cells. So in our previous sections of AP Bio Unit 4, we saw on videos 4.1 through 4.4 that ligands are what cells use to communicate. So oftentimes these hormones are our ligands. And we'll use negative feedback systems to maintain a constant internal condition. So the way the endocrine system works is we have glands that send hormones, release hormones into our bloodstream. And then our bloodstream, so these hormones are going to travel throughout our entire bodies, but they really only have an impact on their target cells. So depending if it's a lipid-based hormone, it'll travel through the blood and enter inside of a cell to attach to its intracellular protein receptor. And if it's a protein-based hormone, such as insulin, it'll travel through the bloodstream and attach to a receptor on the surface. Now, this slide is copied from a previous video. But yeah, the key here is that hormones reach their target cells. They travel long distances by traveling through the bloodstream. Okay, so let's go ahead and see how our endocrine system helps us to maintain homeostasis or constant internal conditions. So our first example we're going to look at is blood sugar or blood glucose levels. So flowing in our blood, we have glucose and red blood cells, white blood cells, etc. So in our blood, we try to maintain a constant level of blood sugar. We call this the set point. So in blood sugar levels, it's around like 90. And so with that, we want to make sure that we always have sugar available for our cells. So let's pretend in this little set of slides, we're going to think about 3 glucose molecules as homeostasis. So let's pretend you eat a high carbohydrate or high sugary meal, right? Maybe it's some fries and a soda, or here, peanut butter and jelly sandwich, and you see the blood sugar levels rise. Now, right here, it's kind of interesting. We can tie this back into a previous concept in biology, dealing with osmosis. So what we just did is we just created a high solute concentration around our blood cells. So now our blood plasma is basically hypertonic in comparison to our cells. So a person with high blood sugar is probably going to feel a bit thirsty as the body craves more water to reach isotonic conditions. Probably going to go to the bathroom kind of a lot too. Okay, so now we are out of homeostasis and our body is like, oh man, way too much sugar. So, how do we do this? So, there's actually, as our blood sugar levels rise, there are cells on our pancreas that are able to detect the high blood sugar, and the pancreas is going to release a hormone. So, the cell response would be to release insulin into the bloodstream. So, we saw here, this is our liver and our muscle cells, represented by that pink box. But here's what's going to happen. The pancreas releases insulin, and as you can see, it travels through the bloodstream and it reaches the target cells of the liver and the muscles. Now, the liver and muscles will respond by taking blood sugar out of the blood and storing it as a polysaccharide called glycogen. So glycogen, if you remember back in AP Bio Unit 1, is the energy storage polysaccharide that animals use to store excess sugar. So our liver can store about 24 hours worth of sugar, which is nice. But we see here in my picture that I mentioned earlier that we're going to use 3 sugars to represent homeostasis. And we can see that our sugar levels are still too high. So now what do? We're not in homeostasis yet. So what will actually happen, because carbohydrates and sugar is made of CH and O, as well as fats, lipids are made of CH and O. So what we can do is we can take that extra sugar and we can convert it into fatty acids or triglycerides and store it in our fat cells. So when people say that carbohydrates make you fat, that is not a true statement. It's only if we take in more energy, more carbohydrates, more sugar than we're actually using, then we're going to store the excess. Think about early humans as hunter-gatherers. If you had a source of energy, you can't afford to just like waste it through like peeing it out or pooping it out. You're going to conserve it and you're going to save it for the days when you can't find food. So by converting our extra carbohydrates into a lipid is a much more compact form of energy, making it easier to carry around with us. And so it's actually a great survival mechanism. The problem is now that if you like sit down and have a couple slices of pizza and a large soda, you just took in like a third of your daily carbohydrates. So nowadays, we're just taking in a whole lot more sugar than we're actually using. Okay, so anyway, so let's go ahead and see what happens. So now we're back in homeostasis. Ta-da! Okay, so now though, let's pretend that you last ate at lunchtime at school and you run cross country. So now you have practice after school and you're running. Well, this is where we use that blood sugar, right? Our mitochondria are going to use it to make ATP and produce energy for us. But now you can see that my blood sugar levels or the blood sugar levels are low. Well, this is where the opposite hormone will be released. So the pancreas will actually release the opposite hormone called glucagon. And glucagon, as we can see, will travel through the bloodstream. It'll reach the target cells of the liver and it will actually cause the hydrolysis of the glycogen polysaccharide. So it's going to hydrolyze or add a water to each of those bonds, sending the sugar back into the bloodstream to raise our blood sugar levels back to balance so that you can keep on exercising. So now if you kept on exercising though and your blood sugar levels were dropping, your body can switch over and begin to use some of that stored fat, those stored lipids, in order to produce ATP and power your run. Okay, so, and then you keep on going. All right, so now let's go ahead and look at this in a diagram form. We're gonna look at 2 slides here that kind of diagram it out for us. So if we maintain our blood sugar, we try and keep it at homeostasis. Let's say after a meal, our blood sugar rises. In response, our pancreas is going to send out the hormone insulin into our bloodstream where it'll target our liver. And then our liver is going to store that extra blood sugar as a polysaccharide called glycogen, which will bring our blood sugar levels back to set point or to balance. Now, sometimes though, you don't eat for a while and you use up that blood sugar. So your blood sugar levels decrease. And then our pancreas will release the opposite hormone called glucagon. And glucagon, not to be confused with glycogen, which is the polysaccharide, glucagon will travel in your bloodstream to your liver, where the polysaccharide, glycogen, will be hydrolyzed or broken down into individual sugars and put back into our bloodstream to maintain our homeostasis conditions. Okay, so these 2 hormones are like antagonistic hormones where they have opposite effects to like bring us back to homeostasis. Now, this is called a negative feedback loop for like a couple of reasons. So when we talk about, I always find that a little tricky to explain negative feedback. And really the idea here is that when you have a set point, like of blood sugar, and you have a stimulus, so a stimulus brings you above set point. So here, this stimulus causes a great deviation or like a great movement away from set point. We want to decrease that. So we want to make it like more negative. We want to bring that back to homeostasis. Or if our blood sugar drops too low, we want to decrease this gap or the like amount of stimulus. We want to decrease it and make it more negative and bring us back to homeostasis. So that's kind of why it's called a negative feedback loop. All right, so now let's look at it in kind of just boxes, and you're free to pause this video and read it in more detail if you need to. But basically, we keep our glucose around 90, hopefully. And so the stimulus in this example would be rising blood sugar. So a stimulus is anything that moves a body condition away from set point. And then the high glucose levels are detected by the pancreas's beta cells. Then the pancreas will release insulin into the bloodstream. The liver and the muscle cells will receive that ligand and take in and store that sugar as glycogen, and that hopefully will bring us back to set point. Now, at times when our blood sugar is dropping, the low blood glucose levels will be detected by the pancreas. It'll release glucagon as the hormone. It'll travel in the bloodstream to the liver, causing the liver to hydrolyze or break down glycogen and send glucose back into the blood, bringing us back to set point. Okay, so now let's go ahead and just see, this is a good time to refresh our memory on cell communication. So here, when we talk about how does insulin work though, again, this is in a whole nother video, I think it's video 4.3. So when we look, 4.3. And when we look at how insulin works, the ultimate cell response is to embed protein like channels or carrier proteins into the cell membrane. So when insulin is received by the target cell in the liver or other cells like muscle cells in the body, one of the responses is to move those protein channels into the cell membrane so glucose can enter the cell from its high concentration to its low concentration. And that's how we take glucose out of the blood. So now let's go ahead though and look at both kinds of diabetes. Like how do our cells like what's happening when we have a what is the word, malfunctioning system, I guess, when we're not able to maintain homeostasis for blood sugar levels. So in the first one, let's look at the left side. In type 1 diabetes, a person's pancreas, the beta cells, are actually attacked by their own immune system. So I believe it's their T cells that attack their own pancreas cells, and their pancreas beta cells no longer release insulin. So even when there's high blood sugar, there's no release of the ligand, and therefore there's no cell response, and the blood sugar levels stay high. Now, in type 2 diabetes, the pancreas works just fine. And as you can see, the insulin was released. However, it's the receptors that basically don't respond to insulin. So if the receptors don't respond to insulin, I think it's like insulin resistance is what it's called. There's still no cell response. The blood sugar levels still remain high. Just the reasons why are different in type 1 or type 2 diabetes. Yeah, okay. So now, though, let's go ahead and look at a few more examples in a little bit more detailed feedback loops involving more parts of the body besides just like one organ like the pancreas. So we're going to look at some examples that connect the brain to the pituitary gland in a feedback loops. I think we have about 3 or 4 examples. Okay, so here we have the hypothalamus. It's in like the center part of our brain. And when we form during development, it's actually pretty rad. When we form during development, there's some cells that form like starting from like the roof of our mouth, and they actually like migrate up towards the brain, and that will actually form the anterior pituitary, which is right below the hypothalamus. And then it's almost like you took a balloon and like stretched out the hypothalamus. You have the posterior pituitary. And so that is more of an extension of the hypothalamus, but these are all located inside of the brain. So now let's go ahead and see how the brain and the nervous system is connected to the endocrine system to help us maintain homeostasis. Okay, so there are some specialized cells in the hypothalamus called neurosecretory cells. So they actually make certain chemicals called neurohormones that will, so like here, these pink little triangles are going to represent these neurohormones. So they're actually made in the brain, and they're going to travel through the bloodstream, but only a very kind of short distance. Their target cells are in the anterior pituitary. So to summarize what we just saw, there's specialized like neurons in the brain called neurosecretory cells that release neurohormones into the circulatory system, into the bloodstream. The blood is going to carry them to the anterior pituitary, where they find their target cells. So these pink triangles are ligands. They're hormones. They'll attach to receptor proteins. So now here we have the target cells. You can see how the ligands will attach. Now their cell response in the anterior pituitary is to send out their own hormones. So here we have some anterior pituitary hormones that will travel in the bloodstream to their target cells. Okay, so now we're going to look at some examples of this relationship. So one of our first hormones we're going to talk about is the thyroxine. And so we have our thyroid gland. It's located here in our like neck region, and it's involved in regulating our overall metabolism. So when we talk about a person having a high metabolism, their thyroid is producing a lot of hormone thyroxine. If their metabolism is low or slow, they're producing less. We want to try and maintain constant balance for our thyroid levels, so that way we are like in a steady state and we're not getting out of balance with hyper or hypothyroidism. So here is the connection. Oops. So the hypothalamus will send out some neurohormones to the anterior pituitary. The anterior pituitary will send hormones to the bloodstream that target the thyroid. And then the thyroid, in response, will send out thyroxine. Okay, so now when we look at this, here, the name of the hormone sent out by the hypothalamus is called TRH, which is TSH releasing hormone. The blue one is TSH, or thyroid stimulating hormone, and the red dot is thyroxine. So here's how it works. So the hypothalamus sends a message to the anterior pituitary. Hey, anterior pituitary, go ahead and release some TSH. So anterior pituitary releases TSH. When TSH travels through the bloodstream and it reaches the thyroid, that is the ligand, and the cell response is to send out and produce thyroxine into the bloodstream. Thyroxine is what helps keep our metabolism levels balanced. So here it does in response. However, if we get a lot of thyroxine, we need to kind of slow this down. So if thyroxine levels increase to too high, then that information, those levels of thyroxine, will be detected by the hypothalamus as well as the anterior pituitary. So that information, that level of thyroxine in the blood, we call it that it feeds back, and the hypothalamus will detect how much thyroxine levels there are, and it will slow down its TRH production. It'll produce less TRH. And if the hypothalamus produces less TRH, then that means the anterior pituitary will respond by producing less TSH, thyroid stimulating hormone. And if there's less TSH targeting the thyroid, well, then there'll be less thyroxine being produced and our metabolism will slow down. However, if these levels get too low, well, then the hypothalamus will increase the amount of TRH, which tells the anterior pituitary to increase its TSH, which tells the thyroid to increase its thyroxine. So they're always like detecting different levels and feeding back to alter how much TRH or TSH are being produced to keep that amount of thyroxine hormone balanced. Now, it's not just the hypothalamus, though, that interprets the thyroxine levels. The amount of thyroxine can also feed back to the anterior pituitary. So I do want to say, though, that when I say feedback, I want you to think about like receptor proteins and the thyroxine being a ligand. Like the anterior pituitary and the hypothalamus will have receptor proteins like receptors for thyroxine. So if their receptors are empty, that tells them, hey, thyroxine levels are low. But if the receptors are keep having thyroxine attached, that also tells them that thyroxine levels are high. So depending on how often thyroxine attaches to the receptors, the hypothalamus and the anterior pituitary can draw conclusions and turn on or turn off different cell signaling pathways to produce the appropriate amount of hormones to try and keep our thyroxine levels balanced. Okay, so if you want to see this written out, so here we have our hypothalamus. It produces the TRH hormone, and then that will target the anterior pituitary. Then in response, the anterior pituitary will produce TSH, which targets the thyroid gland, and then the thyroid gland in response produces thyroxine. So the levels of thyroxine will be detected by the hypothalamus as well as the anterior pituitary, and depending on the levels of thyroxine. So if thyroxine levels are super low, well, then you'd have more TSH and more TRH and more TSH. If thyroxine levels are really high, then you'd have less TRH and less TSH to slow down the production of thyroxine. And that is a negative feedback loop. Okay, so let's go ahead and look at an example of some steroid hormones. So here we have the feedback loop involved in testosterone production. So here, again, it's using the hypothalamus, the anterior pituitary, and the testes to keep like testosterone levels balanced. So here, the hypothalamus will detect, and I believe anterior pituitary will detect levels of testosterone to always make sure that there's sufficient amounts. So what will happen is the hypothalamus will release GnRH, and then it'll target the anterior pituitary. In response, the anterior pituitary will will release LH and FSH. LH and FSH will target the testes, and their cell response will be to release testosterone, and testosterone will travel throughout the bloodstream. So this is how the cells in the body or in the brain can detect and maintain and have an influence on the levels of testosterone, always keeping them at high enough levels to maintain gene expression or sperm production or whatever. So one thing I do want to point out, what was my slide? Okay, so if testosterone levels drop, then you can expect to have high amounts of LH and FSH. If testosterone levels increase, you'd have lower GnRH and lower LH and FSH. Okay, so let's go ahead and look at how you would diagram this out. So here, if you have your hypothalamus, it releases GnRH, targets the anterior pituitary to release FSH and LSH that targets the testicles to release testosterone. Now, depending on the levels of testosterone, that will influence how much GnRH and how much FSH and LH are produced. So I want to point out, if you're sitting here thinking, oh man, this lady is talking way too fast. I'll never remember all of these names. If you are in AP Bio, you don't have to memorize any of the names of these hormones. It's really just how they work and like listen to each other, and how they influence each other. So yeah, you don't have to stress about memorizing, but know how to interpret this. So let's give an example here. So sometimes, like men or women, but we'll go with a male bodybuilder. Sometimes we'll take like anabolic steroids in order to increase muscle mass. And what actually happens if they take like anabolic steroids, they are like steroids that mimic testosterone. So if a person takes steroids, the body thinks that there's a ton of testosterone. And so what will actually happen is the hypothalamus will lower the GnRH, and then it will lower in response, the anterior pituitary will release less FSH and less LH. And the testicles, it's kind of like supply and demand. The testicles are like, oh, we don't need to produce so much testosterone like that we were. And testosterone is involved in making sperm. And so what happens is the testicles will actually shrink quite a bit simply because they're not producing in response to like low levels of FSH and LH. So kind of fascinating. Okay, so let's go ahead and look at ovaries and estrogen in the female. It's the same thing. Females also have the GnRH, gonadotropin releasing hormone, that will target the anterior pituitary, will release LH and FSH, which in females are actually involved in the production of the ovum or the egg. So, like LH is luteinizing hormone, and then FSH is follicle stimulating hormone. And the levels of these hormones are actually going to cause like meiosis to happen, where the egg will like go like be produced, and then you have ovulation where the egg will be released about mid-cycle. And so anyway, so then estrogen will be released in response. But for females, these hormones aren't kept in a steady state throughout her entire cycle. For females, like if we look at, let me just skip over. If we look at a female's cycle, we can see how the, okay, sorry. All right, here we go. So we can look at how these hormone levels can change throughout the month. And so they each play their own role in female reproduction. And depending on the levels of LH, estrogen, and FSH throughout the cycle, will determine when the female ovulates and the egg is released. And then progesterone will affect progesterone levels, which is basically preparing the body for an embryo. Now, I do want to point out, though, if a female takes like birth control is synthetic hormones or man-made hormones, and it like basically tricks the hypothalamus. So if we go back to this, if a woman takes birth control that mimics, for example, LH and FSH, then what happens is that feeds back to the hypothalamus and tells the hypothalamus that there's a lot of this hormone. So the hypothalamus will make less GnRH. And if it makes less GnRH, then it makes less LH and FSH. And then the ovaries won't go through, it won't make an egg. Like there'll be no meiosis. You can see in the beginning of the cycle here, the blue and the green, how there's an increase here. So anyway, this is how the like birth control pills work, by interfering with the, or like mimicking the hormone like feedback loop that happens. Okay, so if we were to read this, here's how it would look, the same as all of the others. So for example, if you are ever reading any of these, you want to think to yourself, okay, so if you increase GnRH, that means you'll have an increase in FSH and LH, and therefore an increase in estrogen. Or if you have a whole lot of estrogen, that'll tell the hypothalamus to slow down and decrease the amount of GnRH, which would decrease the amount of FSH, and LH, which would decrease the amount of estrogen. So the information is detected by different places in the brain. Okay, and our last example of negative feedback is on water balance. So our posterior pituitary that was actually formed from an extension of the hypothalamus, is different than the anterior because here, the posterior pituitary gland doesn't actually doesn't actually produce any hormones. The two hormones that come out of the posterior pituitary are actually made in the hypothalamus. So the two hormones are ADH and oxytocin. We'll talk about oxytocin in our positive feedback loops video, but right now let's go ahead and talk about ADH. So there are sensors in especially in the back part of your brain back here, like in your brain stem that will detect, they're there, but also in your hypothalamus that measure your blood osmolarity. So what is your solute concentration, right? Like, are you hypertonic, hypotonic, isotonic? Like, what is your blood osmolarity? And we want to maintain that constant isotonic conditions, hypotonic, sorry, homeostasis, right? And so we have a hormone for that. So when our blood osmolarity moves out of isotonic, so whether we become hypertonic or hypotonic, we need to check that and regulate. So this is called osmoregulation. So here's what's going to happen. We have this hormone called antidiuretic hormone. And the antidiuretic hormone, basically anti-diuretic, like anti-pee. And so if you are dehydrated and your blood is becoming hyperosmotic, like you have lots of solutes in your blood, then the brain, the hypothalamus, will produce ADH, antidiuretic hormone. It's just released from the posterior pituitary, and it's going to travel in the bloodstream to your kidneys. When it gets to your kidneys, it'll actually change the amount of aquaporins in the collecting duct. So, what will happen is it will help to conserve water and actually bring water in from our filtrate, in the liquid that's becoming our urine. We'll take some water back, basically saying hey, we're hypertonic, we need to conserve water. Now, on the opposite though, if you're overhydrated and you have so much water, you're hypotonic in your blood, then yes, you need to urinate and get that excess water out so your cells don't undergo cell lysis and burst. So therefore, in those conditions, you would not release ADH. We don't want more aquaporins. We want our water to leave our body and bring us back to isotonic conditions. Now, when we talk about in life, certain toxins. So alcohol is a toxin, and one of the ways it causes harm in the body is it interferes with this hormonal pathway. So when a person drinks alcohol, it's a toxin, and it's going to interfere with the amount of ADH that is released. So when a person drinks alcohol, they tend to stop or lower the amount of ADH released, and therefore they continue to urinate and basically dehydrate themselves. And that's what leads to a massive hangover, and the headache is severe dehydration because the hormonal balance was disrupted by the toxin. All right, so that is what ends my long video on negative feedback loops. My next video will be shorter on positive feedback loops. Good job.